So I'm Dinesh, doing my PhD at Leibniz Institute for Agriculture Engineering and Bioeconomy. Today I'm going to present our study on the microbiome of wild flowering plants in the alpine ecosystem, focusing on the distinct microbial communities in flowers and leaves. So to choose alpine ecosystem, this offers a pristine environment with high UV exposure, low temperatures, also limited nutrients. So the microbes here are adapted to harsh conditions, which makes them to be resilient. So our main objectives to focus on understanding how microbial communities are varying between these plant compartments. Especially we focused on flower and leaves. Also, we would like to know how these environmental factors such as location, altitude, and soil type is influencing these communities. We also investigated if plant phylogenetic distance impacts the microbial composition by exploring the concept of phylosymbiosis. For this, we collected samples from 7 locations, so across um, different alpine altitude ranges of 450 meters to 2,200 meters, covering 2 distinct soil types, which is silicate and carbonate soils. So the workflow included DNA extraction, sequencing, and further data analysis to investigate our microbial diversity and composition. So the first result, we were able to see a clear microbial diversity and composition pattern between flowers and leaves. So the leaves always shows a higher diversity than flowers. Also, the distinct cluster between flowers and leaves is shown in the PCOA plot. Further, we looked into Lipsey analysis to highlight specific taxa. For example, in each compartment, such as Pseudomonas in flowers and Spingomonas in leaves. So the lower diversity in flowers may be due to their short lifespan and nutrient rich in nature that makes a fast growing microorganisms to limits the microbial diversity. But in contrast, leaves always provide a stable habitat compared to flowers. This allows the establishment of diverse microbial communities. Moving into our environmental factors, the first one is sampling location. We are able to see a clear diversity pattern on fungal communities, but not in the bacterial communities. Also, the community composition for both bacteria and fungi was significantly influenced by the location. So this explains fungal communities may be more sensitive to location environmental variation factors like temperatures, humidities, and microclimatic variations, but bacterial communities shows a less variation in diversity. This may be due to their strong dispersal abilities and potential ecological tolerances. Moving into our soil type, interestingly, we were not able to see any diversity difference between carbonate and silicate soils, but there is a influence on the community composition between these two soil types. This, the influence are maybe because of soil physiochemical properties. For instance, these particular two soil type may have a contrasting phosphorus availability. This could affect the microbial community assembly directly through plant mediated process. Moving on to our altitude factor, similar to our geographical location, we were able to see the fungal communities were significantly increased with altitude on the diversity side. Also, the community composition were influenced with the different altitude ranges for both bacteria and fungal on both the plant compartments. This also explains this fungal diversity may be adapted to lower temperatures and to low oxygen levels, favoring to psychrophilic fungal communities. But bacteria may be more resilient to the altitude-related factors like temperature and UV exposures. So further, what we did to understand the core microbiome variations across the altitude, so we subset the average altitudes from 400 to 2000 meter altitudes. So as a core microbial classes, and for instance, in fungal communities, we can clearly able to see the class Deuteriomycetes decreasing while increasing the Letiomycetes with altitude. Also, in bacterial side, the Gamoproteobacteria consistently dominated in flowers, while Alpoproteobacteria were prevalent on the leaves. For further, we looked into phylosymbiosis evidence for both flowers and leaves, and we can see a mental correlation significantly between the microbial distance of both bacteria and fungal with their phylogenetic distance. but we were not able to see any significant relation on the leaf side. This explains flowers are reproductive in nature and tend to have specialized interaction with pollinators. Also, it often produces a unique chemical compounds that can select a specialized microbial community that closely align with plant evolutionary histories. So, by concluding this, alpine plants offer us a natural model system for us to study the plant microbe interaction, contributing to the resilience and adaptation of these microbes. This research also helps us in understanding how these microbial communities are responding to environmental changes, such as climatic shifts. So finally, I would like to thank my sincere thanks to my supervisors, Dr. Gabriela Bjerg and Dr. Ahmad Abdul Fattah, for their support and contributions. Thank you so much.